I ended the last lecture by asking what would happen if an infection got out of hand. We saw in that lecture, you'll remember, how the so-called non-specific or innate immune response provides a general defense against injury or infection. But sometimes a pathogen may be resistant to these non-specific defenses or may be able to multiply too rapidly to be overcome by them. In these cases, the more efficient and more targeted mechanisms of the so-called specific immune system, or what we also call the acquired immune system, come into play. In today's lecture, we're going to take a general look at how these mechanisms work. Now, there are two fundamental features that distinguish what we call the specific immune response, or equivalently, the acquired immune response. The first feature is the ability to recognize particular kinds of foreign material in a highly specific fashion, hence the name specific immunity given to this. Now, the second feature is the ability to acquire a long-term memory of exposure to these particular kinds of foreign materials. And this long-term memory permits an even faster and more efficient response to that same pathogen if it's ever encountered again by an individual. Now, these properties of the acquired immune system are mediated by another set of leukocytes. Remember, leukocytes is the term we gave for white blood cells. These leukocytes are called lymphocytes. There are two general kinds of lymphocytes that we're interested in, so-called B cells and T cells. Now, like other kinds of white blood cells, B cells and T cells circulate throughout the body in the blood and the lymph and interstitial fluids of the tissues. But these lymphocytes are most commonly found in organs of the lymphatic system that we discussed last time, especially when they're not at work, which is what gives rise to their general name of lymphocytes. Now, B cells and T cells take their specific names from where they develop in the body. Like all leukocytes, actually all white blood cells, they develop initially in the bone marrow of mammals and other vertebrates. Now, T cells, before they finish their development, travel to a gland called the thymus. This is a gland that's just over the heart in a typical vertebrate body, and that's where they get their name T cells. In humans, B cells start and finish their development in the bone marrow. Actually, you would think then that B cells might stand for bone marrow cells, but actually, the B comes from the term bursa, which is a special lymphatic organ found only in birds. Now, you might ask, why are we naming this after birds? And the reason is because B cells were originally discovered long ago from experiments done on birds. So the B comes from bursa. But we can remember B cells as bone marrow cells for our purposes, and T cells as cells that develop in the thymus. Now, B cells and T cells are the principal players that we need to keep track of, but what do these cells do? Now, in response to the invasion by a pathogen, in the body, the B cells are the cells that will release copious amounts of proteins called antibodies. You may have heard this term before. Antibodies are proteins that will bind in a specific fashion to foreign material, and in so doing, help remove or destroy it by mechanisms we'll discuss in a little while. This antibody response mediated by the B cells is sometimes called the humoral component of acquired immunity. Humoral, in this case, hearkening back to an archaic term for body fluids, because this response is doing its work largely in the body fluids of the organism. Now, there are several kinds of T cells that we'll encounter. We really only need to consider two of these to understand how specific immunity works. One kind of T cell, which we're going to call helper T cells, actually play an essential role in initiating the acquired immune response, and we'll talk about that a little later in the lecture. Now, the second kind of T cell, which are called cytotoxic T cells, this is another one of those killer names, actually play a more direct role in defense. Their role is similar to those natural killer cells that we talked about last time, in that these cytotoxic T cells actually directly attack cells that have been invaded by viruses and kill them. The action of T cells is sometimes referred to as the cell-mediated component of the acquired immune response, because these cells are actually doing the work. Now, here's the point we need to get to. Both B cells and T cells are characterized by having receptor proteins on their cell surfaces that recognize foreign materials in a highly specific fashion. It's these specific receptor proteins that account for the ability for these cells to recognize particular kinds of foreign material in a highly specific fashion. Now, I need to define the word antigen to make this point clear. Now, by definition, an antigen is any molecule that triggers the acquired immune response. The term actually is short for antibody generator, antigen, and it was given these proteins were given antigens were given this name because early on in studies of the immune system, the expression of antibodies was the most obvious thing that people could measure about how the immune system worked. So the term antigen simply refers to any molecule that's recognized as being foreign to the body. But of course, there are many, many such molecules, so there can be many, many kinds of antigens. And any particular pathogen, such as a bacteria, will almost inevitably have some, and actually probably many, many molecules on its surface, such as membrane proteins and whatnot, that will be recognized as antigens. Now, there are a couple of key points to emphasize. First, any one lymphocyte, that is, any particular B cell or T cell, will only have receptors that are specific for a single kind of antigen. In other words, one cell will only recognize one kind of antigen because it only has receptors that recognize that antigen. But the second point is that in a given individual, there are actually potentially millions and millions of different cells that have different antigen receptors. What that means is that in aggregate, some cells in the body of an individual are likely to recognize just about any antigen that's encountered. Now, the claim that I just made, that each lymphocyte can respond only to one antigen, but that in fact there are many, many kinds of lymphocytes with different antigens, actually gives, with different recognition receptors for different antigens, actually leads to two puzzles that we want to address. The first puzzle is, how is it possible for lymphocytes in the body of an individual to have so many different kinds of antigen receptors? These antigen receptors are proteins. And from what we've learned about the way proteins work, what this must mean is that each antigen receptor is a different kind of protein. Because the different kind of protein, the different protein composition will have a different shape, and that's how we know proteins recognize other molecules in a highly specific fashion. Now, it seems very unlikely that there could be a different gene for every different kind of antigen receptor because we'd have to have millions and millions of different genes. So the first question we want to think about is, how is it possible to have so many different receptor types given what we would expect to be a more limited set of genes? Now, the second question that we want to address, the second puzzle really, is that if there's only one or maybe a few lymphocytes in any individual's body that have antigen receptors that recognize a particular antigen, then how does this few number of cells actually mount an effective response? We would expect the response to be maybe an overwhelming response, but only a few cells might have a particular antigen receptor. Let's consider this second puzzle first before we go back to the first. Now, when a lymphocyte encounters an antigen that actually matches up with its receptors, that lymphocyte will generally be triggered to divide very rapidly, producing many, many daughter lymphocytes that all have the same antigen receptor. Now, the details of how this interaction between antigen and lymphocyte occurs can get quite complicated, but let's ignore the details for the moment and consider just the big picture. Remember, I said at any one time, there may be only a very small number of lymphocytes having a particular antigen receptor that recognizes a particular pathogen, particular antigen on a pathogen. But when one of those cells does encounter the right kind of antigen, the matching antigen, it produces a clone. Remember, a clone is just a name that refers to cells having an identical genetic makeup. So that means in a very short period of time, there will be a huge number of lymphocytes that all have the same receptor type, the receptor type that matches the antigen that's been encountered. And now all of these cells can participate in eliminating that particular pathogen. This production of lymphocytes in response to an encounter with an antigen is called clonal selection. It's called clonal selection because only the few cells that have the correct antigen receptor are going to be selected to produce clones. Now, clonal selection easily solves the puzzle of how only one or a few cells having a receptor that recognizes a particular antigen can go on to mount an effective defense. Now, it turns out that clonal selection also accounts for the ability of the immune system to retain a memory of particular pathogens that it encountered earlier in the following way. When a lymphocyte, either a B cell or a T cell, is stimulated to produce a clone, it actually produces two kinds of cells. It produces one kind of cells that we generally call effector cells. These are the cells that are going to actually go on and participate actively in the immune response. These are the cells, the B cells, that will go on to actually produce antibodies, and the T cells that will go on to actually attack infected cells. But these clones will also include another kind of cell called memory cells. Now, memory cells don't actually participate in the ongoing attack against the pathogen. In effect, they actually just sort of sit back and wait to fight another day. They get stored elsewhere in the body, and these memory cells can last for years or even decades. What that means is that once the body has encountered a particular antigen, certain cells will recognize that antigen sometime in the future. And because these memory cells are stored and waiting, the body will be able to respond much more rapidly and, in fact, much more efficiently the next time it encounters the same antigen. Now, you can actually demonstrate the role of memory cells in this kind of response if you measure, for example, the concentration of antibodies that are found in the blood when an individual encounters an infectious pathogen. You can measure these antibodies a number of ways, and I won't go into the details, but if you measure antibody production, what you would find is that when an individual encounters an antigen, a pathogen, for the first time, over a time course of maybe a week or two, the level of antibodies that are specific for that pathogen will mount steadily in the bloodstream. And this is because these antibodies are being generated to fight off that infection. And then they'll gradually decrease over another week or so as that infection subsides. Now, if you take that same individual and expose it to the same antigen at some future point in time, what you find is that the production of antibodies occurs much, much more rapidly. And in fact, many more antibodies are produced. You have a higher concentration of antibodies. It's this so-called secondary response that results from the original production of memory cells that remain in the body and wait in case there's a second encounter of the same antigen. Now, there's another puzzle that I haven't mentioned yet about how acquired immunity works that may not be apparent, but I want to address this one now. I told you that lymphocytes, when we consider them in aggregate, can have antigen receptors that are specific for millions and millions of different kinds of potential antigens, and we still haven't accounted for how we can generate this diversity. But first we want to ask the following question. If the antigen receptors can be directed against so many different kinds of molecules, what prevents there from being lymphocytes that have basically receptors that recognize molecules of the individual's own body and thus triggers an immune response to them? In other words, what ensures that there are only antigen receptors found on lymphocytes that recognize non-self materials and avoid recognizing self materials? And of course, this is critical. Otherwise, lymphocytes would initiate attacks against normal, healthy cells and tissues of the individual's body. Interestingly, the answer to this question appears to be that lymphocytes, actually not only appears to be, I should say, is that lymphocytes with antigen receptors recognizing self molecules are produced all the time. They're developing all the time in your bone marrow. But they're eliminated before they complete their development. In essence, what happens is this. The young lymphocytes, as they're developing, if they happen to have receptor types that recognize self molecules, will start responding to those self molecules because, of course, they're developing in the tissues of the individual's body, so they're going to be encountering self molecules all the time. Now, these lymphocytes will be responding in a way that's not sufficient to trigger an immune response, but which nonetheless triggers a cascade of cellular events in that cell. This cascade of cellular events, essentially a weak response to self molecules, triggers a set of signals from the outside to come in and tell that cell to destroy itself, or otherwise turn that cell off so it no longer develops. So what's really happening is that these lymphocytes are being tested constantly during development. And if they test positive for receptivity towards self molecules, they're simply eliminated. In fact, one estimate has about 90% of all lymphocytes produced in the body being eliminated this way, with only the remaining 10% having antigen receptors that recognize foreign types of antigens, and thus going on and doing the work of the acquired immune system. Okay, now what I want to do is to turn to that first puzzle I mentioned, which is how an individual can produce lymphocytes having so many different kinds of antigen receptors. It's estimated, for example, that B cells in a typical human individual can potentially respond to somewhere between several million to a billion different kinds of antigens. This is a huge number. How can an individual produce different proteins with this much variability? Now, there is one easy possibility, and that is that there are millions of different genes coding for millions of different protein types. But we can rule this hypothesis out rather quickly if we just do a rough estimate about how much DNA that would take. In fact, the estimate comes in so large that the amount of DNA necessary to account for that kind of variability would more or less take up the whole genome of the individual, and there'd be no room for anything else. Now, an alternative possibility for how this could work was proposed in the 1960s by W.J. Dreyer and W. Claude Bennett. Now, their idea was controversial at the time, but after many years of research, it's proven true. In a nutshell, their suggestion was that the genes responsible for producing antigen receptors basically come in parts. And in fact, that these parts have many different versions that become shuffled permanently as a lymphocyte develops. Now, this idea was controversial because it suggested that genetic recombination is occurring in a cell. Now, we've seen genetic recombination before. Remember, when we talked about the process of meiosis, we saw that genetic recombination could occur through crossing over. But what we're talking about here is a fundamentally different process, because this would be a cutting and pasting of the DNA of an individual cell outside of sexual reproduction. It had never been observed before, and in fact, it only occurs in these cells that are producing lymphocytes. Now, even if you only have a relatively small number of parts, versions of parts that you shuffle this way, you can imagine that you could easily generate a huge number of combinations. Now, to understand this in a little more detail, let's look at what these antigen receptors really are, very briefly. And what we're going to do is we're going to consider B cell receptors. These are the antigen receptors that occur on B cells because they're the best understood. The antigen receptors on B cells are members of a family of proteins called immunoglobulins. Now, actually, B cell receptors are really a type of antibody. Antibodies come in a number of different forms characterized by sort of a different tail region to the molecule, and B cell receptors just happen to be a form of antibody that has part of the molecule that allows it to embed in the cell membrane and thus be a receptor on that cell membrane. So we can think of B cell receptors and antibodies in more or less the same way, at least from the point of view of their molecular structure. Now, B cell receptors, and this is the point, are composed of several parts which are functionally and structurally distinct. First of all, the receptor is actually composed of 4 different proteins that are combined together. There are 2 identical shorter pieces that we'll call light chains, and 2 identical longer pieces, longer proteins, that we'll call heavy chains. Now, these 4 parts, these 4 proteins, come together in sort of a Y-shaped structure. You can imagine this, picture this as sort of 2 long proteins that are connected together along one half, and then on the other half they split apart, forming a Y-shaped structure. Now, those are the heavy chains that we're talking about. Now, on the arms of the Y, combining with the heavy chains are these 2 shorter light chains. Now, the important point here is that is not the overall structure of the receptor, but the fact that different parts of this receptor are variable in different ways. And specifically, for each of these light and heavy chains, there's what we call a constant region to the protein, which is the same for every kind of B cell receptor. And then there's a variable region. The variable region is the part of these proteins that is occurring in the outermost part of the arms of the Y. In other words, the part of the receptor that's sticking out from the cell. Now here's the point to remember, and that's this, that the variable regions of these proteins are what come together to form the binding site, the specific binding site that recognizes a particular antigen. Now, T cell receptors we're not going to talk about. They're structurally very different from B cell receptors, but the same thing occurs. There is a constant region to the proteins that make up the T cell receptors and a variable region. Now, given this background on the structure of antigen receptors, let's look again at how we can generate so many different kinds of receptor types. Let's start by considering just the genes that code for the heavy chains of B cell receptors. There are actually 4 sets of genes that contribute to each individual heavy chain. Now, 3 of these sets of genes will contribute part of the sequence that codes for the variable region of the chain. The 4th set of genes will code for the part of the sequence that we call the constant region. Remember, it's the huge amount of variation in the variable region that we're really interested in accounting for. Now, when a stem cell in the bone marrow begins to differentiate to produce a mature B cell, that cell's DNA gets cut up and pasted back together in such a way that only one version of each of these 3 sets of genes will contribute to the variable region of the one protein that actually now is the only protein that can be coded for by the genome of that cell. Imagine this as follows. The cell actually starts with a very long stretch of DNA that carries many different copies of each of these same 3 kinds of segments that will ultimately add up to produce the variable region. In fact, there are about 100 or so versions of one kind of segment, about 30 or so versions of another segment that's going to contribute, and then about a half dozen versions of a third segment. Now, putting these together is a little bit like ordering from a restaurant menu where you can have one from column A, one from column B, and one from column C. As the DNA is cut and pasted together, only one version of each of these segment parts is retained. All of the rest are eliminated from the DNA of that cell. And these versions of each segment then are pasted back together. And now the genome, the DNA of that cell, has one continuous stretch that codes for a unique protein, a protein that has these 3 segments just now contiguous in coding for the amino acid sequence of that protein. You can imagine with 100 versions of one segment, 30 versions of another, and 6 versions of the third, you can produce actually millions of different kinds of combinations. And if you add to that the fact that the light chain, which is going to also contribute to the variable region of the antigen receptor, also has a recombination of its DNA in this way, and we can combine the light chains and heavy chains in different combinations, the number of possibilities becomes truly astronomical. It's this genetic cutting and pasting that ultimately is what's responsible for creating all of the variation that's necessary for the acquired immune response to be able to have receptors that can respond specifically to so many different kinds of antigens. Okay, now there are a lot of other aspects about the way that acquired immunity works in vertebrates that we really haven't touched on yet. It's a very complicated and fascinating system, and fortunately for us, it works really, really well. But the one part of this system that I want to end on is to talk about how the whole system gets initiated in the first place. Now, I've told you already, for example, that when a B cell encounters an antigen, it will form a clone that includes both memory cells and effector cells. These effector cells, by the way, are called plasma cells or plasma B cells, and those are the cells that secrete antibodies. These antibodies have the same variable region, thus the same antigen specificity as the receptor of the B cell, and thus these antibodies will bind to whatever antigen that B cell is responding to. And the way that these antibodies actually contribute to defense is by binding to whatever pathogen they're targeting, and in so doing, making it easier for other parts of the immune system to recognize those pathogens. In other words, when antibodies bind to a bacteria cell, then the macrophages we talked about last time will actually have an easier time recognizing, finding, and engulfing that bacteria. Similarly, the complement proteins that we talked about last time will be recruited more quickly to a bacterial cell, for example, that has antibodies. So essentially, binding to it. So essentially, the antibodies are labeling particular substances as being subject to attack. And in making these labels, they're allowing other aspects of the immune system to come in more efficiently and in a more targeted fashion. Now, when T cells, as I say, encounter an antigen, they also form a clone. Some of these clone cells are memory cells as well, and others are these cytotoxic cytotoxic T cells, I should say, which will go in and target and attack cells that have been infected by viruses, or alternatively, they may also attack cells that have begun to show abnormalities associated with the development of a cancerous condition. Now, what that means is, again, to a first approximation, these effector T cells, the cytotoxic T cells, are actually responding in a way that's similar to what we saw with the nonspecific immune system, but in a much more targeted fashion. But the story with T cells is actually more complex than this, because one kind of T cell that's produced when a T cell produces a clone, what we called helper T cells, also serve to turn on and regulate the entire acquired immune response. How does this happen? Remember I told you last time that those leukocytes we called macrophages will engulf any foreign material and digest it. It turns out they do more than that. They not only will digest foreign material, say a bacterial cell, but they'll also process parts of that cell, parts that are antigens, combine them with a special set of proteins called MHC proteins, that stands for major histocompatibility complex, and then they'll present the antigen and MHC protein on the surface, on their own cell surface. These macrophages then travel through the lymphatic system to lymph nodes where they encounter T cells. T cells will only be triggered to produce a clone if they encounter the right antigen when it's been presented with an MHC molecule, an MHC protein, by a macrophage. So they don't just respond to these antigens on their own. They need to be presented by a macrophage, which is digested, processed, and presented the antigen. Now, when these T cells form a clone, these helper T cells now have a specificity for this antigen, but they will now go out and not necessarily encounter antigens on their own, but instead look for other cells that have processed and presented those same antigens. Specifically, B cells, those are the cells that produce antibodies, when they encounter an antigen, will actually take some of that antigen in, combine it again with a kind of MHC protein, and present it on their surface. Now, the B cell itself won't be triggered to do its work. It won't be triggered to form a clone and to produce these effector cells, the plasma cells, until it encounters a helper T cell that also has a specificity for the antigen that it's presenting. So the helper T cell has to come out and essentially handshake with a B cell that has encountered the same antigen. And when that happens, the B cell becomes activated and the helper T cell becomes activated to start producing signals that initiate other parts of their immune response. Now you might ask, why is it so complicated? We've got work to do here. Well, the reason is this, it's thought. The acquired immune response, the specific immune response, is potentially so powerful that it can actually be quite damaging. And so you might think of it like a very powerful weapon in possession of a nation. Generally, those weapons aren't deployed unless the chief executive makes the order. This would be the helper T cell, has to come in and say, okay, it's time. And they won't be deployed unless there's a series of agreements and double checks and handshakes that says, yes, we all agree it's time to do this. So too, the activation of the immune system requires these interactions between different cell types, all of which have encountered the same kind of antigen, processed it in certain ways, and have had a chance to interact with each other before the helper T cell will eventually begin sending signals to say, now it's time. We have to activate the full range of our defenses against this particular invader. An invader that they can now recognize in a highly specific fashion by the particular antigen molecules that it carries.